So, yesterday, wait a second, I'm going to close the door. We do have a private lesson. So, okay, let's just recap a little bit the last lesson. We finished with the with the different steps, okay, during the muscle contraction. So the different steps are the events at the neuromuscular junction, okay, with the release of neurotransmitter, such as the acetylcholine neurotransmitters that bind to the acetylcholine receptor, that in this case is a nicotinic receptor, okay, an ionotropic receptor. And then the second step is the excitation contraction coupling, the EC coupling. Wait a second. Okay, you are right. You are right. Just stop me by using the microphone next time. Tac, tac, tac. Okay. We were saying that we will start to talk again about this one, okay. Good morning. Different events. So the first event at the neuromuscular junction, then the excitation contraction coupling, and then the contraction or relaxation cycles. So we know everything about the events of the neuromuscular junction. As we said before, the acetylcholine is released and it binds to nicotinic receptor that are ionotropic receptor that permit the sodium ions to flow inside of the cell. And then we have to understand how the excitation, contraction, coupling takes place. So how the muscle can contract after the events at the neuromuscular junction. And as we described in the lessons of yesterday, of I don't remember when, Tuesday, okay, we realized that the band A, this one, okay, in these bands, the length is always the same, even if the muscle is relaxed or contract. So when the contraction occurs, the sarcomere from one Z line to the other one at each ends, okay, moves closer together, while the I band, one and this one, and the H zone, this one, almost disappear. The two filaments of actin and myosin overlap, and the muscle contraction can occur. But again, despite shortening of the sarcomere, the length of the A band remains constant. And these changes explain perfectly the sliding of the thin filament along the thick filaments, since the actin moves toward the M line in the center of the sarcomere. Just moving on the right of this figure by the cross bridges that occurred between myosin and actin. We will analyze again the process in a more detailed way in the next feature, okay? Now we know that this process can occur because the thin filaments are able to move along the thick filaments. Let's see this short video.
To start, let's magnify a myofibril and examine the features of the sarcomere. Each myofibril is made up of thousands of sarcomeres. The sarcomere is the basic unit of muscle contraction. By understanding how one sarcomere shortens, we can understand how the simultaneous contraction of thousands of sarcomeres along hundreds of myofibrils leads to contraction of one muscle fiber. Each sarcomere consists of an arrangement of thick and thin filaments. The thick filaments are made up of many myosin proteins. Each myosin molecule is shaped a bit like a golf club, with two heads and a tail. Each myosin head contains two important binding sites. One site binds to ATP, which will supply the energy used for muscle contraction. The second site binds to the myosin binding site on actin. The result is a cross-bridge between thick and thin filaments. Now that we've examined the thick filaments, let's review the structure of the thin filaments. Recall from the IP 2.0 topic, excitation-contraction coupling, that the thin filaments are composed of three types of molecules: actin, tropomyosin, and troponin. The actin molecules form a pair of strands that wind around each other. Each actin molecule has a myosin binding site to which a myosin head can attach. When the muscle is not being stimulated to contract, these binding sites are covered by tropomyosin. However, the myosin binding sites are exposed when calcium ions bind to troponin during excitation-contraction coupling. Now that we have identified the main components of the thick and thin filaments, let's examine how they are arranged within the sarcomere. When viewed with a microscope, the skeletal muscle tissue has a striped or striated appearance with alternating light and dark bands. Let's examine what causes these stripes. The lightest areas are called I bands. The only filaments in the I band are thin filaments. A Z disc in the center of each I band anchors the thin filaments. The Z discs also mark the ends of the sarcomere. The darker central areas of the sarcomere are the A bands. The A band is the region occupied by thick filaments. The thick filaments are anchored to the M line, which marks the center of the sarcomere. The central region of the A band is slightly lighter than the rest of the A band because it contains thick filaments, but no thin filaments. This region is called the H zone.
Okay, we just see the organization of the sarcomere and what happened in the muscle when the contraction takes place. Okay, and this slide just resumes all the process that we discussed until now about the contraction and the relaxation. But what about now the sliding filament mechanism of contraction? Let's check also this video.
Now that you know the regions of the sarcomere, let's examine the sliding filament mechanism of muscle contraction. Recall that the thin filaments extend from the Z disc toward the center of the sarcomere, overlapping partially with the thick filaments. According to the sliding filament mechanism, a muscle contracts because the thin filaments slide past the thick filaments toward the center of the sarcomere from both sides. In doing so, they pull the Z disc at either end of the sarcomere toward the central M line, shortening the sarcomere. This occurs at all the sarcomeres along the length of the muscle fiber. Let's look at how the bands change during muscle contraction. Notice that the I band shortens as the sarcomere goes from a relaxed to a contracted state. However, the width of the A band remains the same length. This is because the filaments themselves do not change in length, they just overlap more. Now let's focus on the H zone. Like the I band, the H zone narrows and may even disappear as the thick and thin filaments overlap more and more. How does the sliding movement occur? During muscle contraction, when myosin heads bind to actin, they form cross bridges. Once cross bridges form, myosin pulls the thin filaments toward the center of the sarcomere. The pulling motion of the myosin heads is similar to how the oars of a rowboat work. The thick filament is the boat, and the oars are the myosin heads that form cross bridges. Each stroke produces only a little movement. However, just as repeated oar strokes move a boat, so the repeated breaking and reforming of myosin cross bridges pulls actin toward the M line. A single action potential is sufficient to stimulate many cross-bridge cycles. This shortening takes place in all the sarcomeres along the length of each myofibril, and in all the myofibrils of a muscle fiber. The result is muscle contraction.
Okay, so we have seen all the process from a structural point of view, okay, how the contraction takes place. And now we are able to understand what happened from the beginning till the end of the contraction, okay? If you remember at the beginning of the course, we analyzed some pictures like this one, okay, when you can see a stimuli, that is an event at the neuromuscular junction, okay, and what happened in the muscle. Here you can have the excitation contraction coupling, the calcium signal, and the contraction relaxation cycle. And we know that during the contraction relaxation cycle, the A band remains at the same dimension. But the filaments just move in a sliding dependent manner. And in the next few slides, okay, we will understand how the sliding filament theory of the contraction takes place. Okay, how the cross bridges between actin and myosin in the actin-myosin binding site allow the muscle to be contracted. The starting point is actin in red here and myosin in violet here slide past each other during the contraction. As we already described and saw in the video, in the two videos before, in the sarcomere there is an overlapping actin and myosin myofibrils. They can have a binding between the actin and myosin filaments. The sliding past each other mechanism is an energy-dependent process, and which is the molecule that per, let's say, default gives energy. It's always the ATP, okay? This process can occur because the ATP is always present in the muscle. The energy molecule is the ATP. So how does the entire process take place? And the next slide, this one, is called the contraction cycle. This process describes how actin and myosin can perform the contraction of a muscle. We already described the myosin heads with the different binding site for ATP and for actin. And we already also described the actin here, the 1, 2, 3 globular actin. We already described that in this molecule is present an actin binding site for the myosin. And in the myosin, there is present an actin binding site in the myosin. So they can, the myosin head bind to actin molecule in binding site. It is the rotation of the myosin cross bridges that provides the force to move actin filaments during the contraction. Let's see the entire process. Okay, let's see what happened in the entire process. As you can see here, the first step here is called the rigor state. Obviously, we are not in the rigor state, fortunately, okay? So, but let's start from here, where the myosin, okay, is bind to actin in the point 1, in the actin number 1, okay? So, this is the rigor mortis state, in which actin and myosin are tightly bound and no ATP or ATP are present, okay? Again, this is not our case. We are not in this case, okay? When the ATP is present, it binds to the myosin head, where? In the ATP site, here. And since it is an ATPases protein, so the myosin hydrolyzes the ATP into ADP and inorganic phosphate. The myosin start to hydrolyze the ATP, and after the attachment of the ATP to myosin, the myosin releases the actin. You can see here. The hydrolysis of the ATP after the release of the actin position 1 allows a weakly attachment to the actin in position 2. You can see here, 1 and 2. The important point is that if ATP is hydrolyzed, both ADP and P remain bound to myosin, as the energy released by ATP hydrolysis rotates the myosin head here until it forms a 90-degree angle. Here is 45. Now here the angle degree is 90, a cocked position with the long axis of the filament. Here you can see the degree from 1 to 2. And the hydrolysis of the ATP, even if the ADP and P are not released, they are still bound to the myosin head. We have here a lot of energy, potential energy, that can be released. Okay? So what happened after that? After that, for a process that we analyze later on, the calcium signal, a calcium signal coming from the endoplasmatic or sarcoplasmatic reticulum, start a process that is called power stroke. Okay? The calcium signal initiates a central process in the skeletal muscle contraction that is called power stroke. So the myosin cross bridges in position 2, swivel and push the actin filaments toward the center of the sarcomere, like a stroke, like a movement of the filament, of the actin filament in the middle of the sarcomere. So this 90-degree angle is full of energy from the ATP ADP. So at the end, when the calcium signal increases, the actin filament is pushed in the middle of the sarcomere, in the center of the sarcomere. The myosin cross bridges here swivel and push the actin filaments toward the center of the sarcomere. At the end of its power stroke, each myosin head releases the actin at this point in position 2, and then swivel back to bind another new actin molecule. And in this case, ADP is released, P is released, and in this case bind to the actin molecule number 3. And when the actin molecule in the position number 3 bind, when the ATP is present, just the process starts again. Always it's important that in the muscle ATP is present and calcium is present. In this way, the power stroke repeats many times as the muscle fiber contraction. Bind, push, bind, push, bind, push. And the muscle can be contracted when the ATP is present and the calcium signal is present. Obviously, the ATP is always present in the muscle. The calcium ions not always are present in the cytoplasm of the muscle. Again, remember, few numbers, but these numbers we need to know. How is the concentration of calcium inside of the cell? How is the concentration of calcium outside of the cell? Always the same question. I know that I'm bothering you a little bit, but now, some numbers are important to remember. You can see here the release of inorganic phosphate allows the myosin head to swivel and move along the axis. The myosin head and the hinge region here, you remember the hinge region here, that are able to form the angles here 45 and here 90. So the release of inorganic phosphate allows the myosin head to swivel and move along the axis. At this moment, the myosin head and the injury region here move from 90 to 45 again. And here, 45. The ADP is released, the myosin head is tightly bound to the actin in position 2, and then we return in the rigor state, but in the position 2. When the ATP is present, we start again the process. Obviously, we are not in this state, okay? The states that in rest condition we are physiologically are this one and this one. So this is the contraction cycle, the contraction and relaxation cycle, okay? But the myosin cross bridges move actin filaments and the muscle is contracted. But as we said before, the ATP is always present in the muscle. But we are not always contract, obviously, even if the ATP is always present. What is important for the contraction? What is necessary for the contraction? Calcium ions. What is the concentration of calcium ions inside and outside of the cell? I'm just repeating some questions that I can ask you during the exam. Actually, I already set up the day for the exam. You will find it in the Al-Maizami webpage. Okay? Let's check after the class. Let's check it. So, resume of the cross bridges, and if ATP is always present, what is necessary for contraction? Now we know that is necessary calcium. We know from the video that we have seen before, but we know it also because we already described that the calcium is necessary. But how, this is again a resume of the things that we have already analyzed, but now the question is how an ion like calcium can allow the contraction. How can switch the contraction on a calcium ion? And the physiological process is this one. If you remember from the beginning of the muscle structure that we have studied, okay, a few lessons ago, we have considered two regulatory proteins, the troponin here and the tropomyosin here, okay? The troponin is bind to the tropomyosin, and the tropomyosin, if you remember, blocks the two binding sites, the one on the myosin head and the one on the actin. The troponin role is to control the positioning of an elongated protein polymer such as the tropomyosin. In the relaxed state here, myosin head cocked. The tropomyosin partially blocks the binding sites on the actin and on the myosin. Myosin is weakly bound to the actin, but since the tropomyosin is here, is blocking the perfect binding of the two proteins, blocks the contraction. But now, when calcium is present inside of the cell, when the calcium concentration rises inside of the cytoplasm of the muscular cells, what happens? The increase of cytosolic calcium permit the contraction. The calcium binds in which way? The calcium binds to the troponin. The troponin with the calcium just shift the position of the tropomyosin. When the calcium binds to the tropomyosin shift the position, move the position, exposing perfectly the binding site on the actin. So the myosin can fully bind to the actin, and the process that we analyzed before can occur. The troponin calcium complex pulls the tropomyosin away from the actin binding site, actin myosin binding site. The myosin binds strongly to the actin and completed the power stroke that we analyzed before. And the actin just move to the center of the sarcomere. In the resting skeletal muscle, the ATP is always present, but lack of calcium. The tropomyosin here wraps around the actin filaments and cover the actin myosin binding site and blocking the two sites. And the myosin in this case is blocked from completing the power stroke. But now, when the calcium increases, it can bind to the troponin, shift the tropomyosin, and the power stroke can proceed. This off-on position of tropomyosin, of a regulatory protein that is the tropomyosin, is regulated by calcium and by troponin, that it's able to bind to calcium reversibly, to bind to calcium. When the contraction begins, calcium can bind to troponin, and the complex troponin calcium pull down the tropomyosin, move the tropomyosin completely away from the actin myosin binding sites. So this on position, calcium dependent, enables the myosin head to form strong high force cross bridges and carry out their power stroke, moving the actin filament in the middle of the sarcomere. Contractile cycles repeated as long as the binding sites are uncovered, as long as the calcium ions are in the cytosolic part of the cell. What is important for muscle relaxation to occur? That the calcium concentration in the cytosol must decrease, and rapidly. But we already know perfectly how the calcium ions can decrease inside of the cell. We have already studied this part in the transport across the membrane, the calcium pump or the active transport, second type of active transport. Remember. So we know now that the the entire process can occur because calcium is present, ATP is present, calcium binds to troponin, troponin pulls the tropomyosin from the myosin binding site on actin, the myosin binds tightly now and moves the actin in the center of the sarcomere, and repeated as long as binding sites are uncovered and obviously ATP is available. Let's see the cross-bridge cycle in this video.
Now that we know that thin filaments slide over thick filaments during muscle contraction, let's examine how the process works in detail. Myosin molecules have two important features that enable the thick filament to bind to and move the thin filament. First, the hinged portion of the tail allows the myosin head to reach the binding sites on actin. Second, the myosin head can pivot back and forth between high energy and low energy positions. This pivoting motion constitutes the power stroke that pulls the thin filament along the thick filament. A sarcomere shortens when myosin heads on the thick filaments form cross bridges and pull on the actin molecules in the thin filaments. The process known as the cross-bridge cycle consists of 4 steps. 1. Cross-bridge formation. 2. The power stroke by the myosin head. 3. Cross-bridge detachment. And 4. Reactivation of the myosin head. Let's examine each step. Step 1. Cross-bridge formation. The cycle starts when excitation-contraction coupling causes the myosin binding sites on actin to be exposed. Myosin heads bind to these sites, forming cross bridges. Myosin starts in its high energy state, in which the ATP binding site on the myosin head is occupied by ADP and inorganic phosphate. The myosin head is ready to pivot for the power stroke. Step 2 is the power stroke. Release of inorganic phosphate from the myosin head causes the head to pivot, pulling the thin filament toward the center of the sarcomere. ADP is then released as well. The myosin head is now in its low energy state, but it remains attached to actin. The energy for the power stroke is ultimately provided by the ATP that binds to the myosin head. Step 3 is cross-bridge detachment. Myosin remains attached to actin until an ATP molecule binds to the empty ATP binding site on the myosin head. This weakens the link between the myosin head and actin, and the myosin head detaches. The detached myosin head remains in its low energy state. Step 4 is the reactivation of the myosin head. This occurs when the ATP bound to the myosin head undergoes hydrolysis to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, pivoting it back to its high energy state. The cross-bridge cycle starts again when the energized myosin head forms another cross-bridge. As long as the myosin binding sites on actin remain exposed and ATP is available, the cross-bridge cycle repeats, pulling the thin filaments toward the center of the sarcomere. During muscle contraction, cross-bridge cycles occur between the thick and thin filaments along the entire length of each sarcomere. But not all myosin heads are bound or disconnected at the same time. They can be in different steps of the cross-bridge cycle. Remember that there are thousands of sarcomeres in a myofibril and thousands of myofibrils in each muscle fiber. Cross-bridge cycling at all of these sarcomeres causes the entire muscle to contract.
Okay, and now the next point is to understand how the entire process occurs. It means the calcium, how the calcium is released inside of the cell. We need to know now this important point, how the calcium signal inside of the cell can permit the contraction relaxation cycle. Because until now, we know everything about the contraction, okay, the red box here. But to understand where the calcium comes from, we need to talk a little bit more about of the somatic motor pathway. We know the somatic motor pathway, which control the skeletal muscle that differ from the autonomic pathways, both anatomically and functionally. And we have already introduced this point and discussed about the single motor neuron and the skeletal muscle in the neuromuscular junction. You remember, or neuromuscular synapses. But we need to know now how the release of acetylcholine on the nicotinic receptor release or in some way allow the calcium signal inside of the cell. We perfectly know here how the signal move from the central nervous system to the end of the motor neurons. And we know how the acetylcholine is released. We know also the point too, how the acetylcholine can bind to the nicotinic receptor. We know also how is the structure of the nicotinic receptor. And how it works, because it is a ionotropic receptor, okay, that allow the sodium ions to move inside of the cell and produce a depolarization of the target skeletal muscle. But now, how is linked the depolarization of the neuromuscular junction and the release of calcium? And this is the point that we are going to discuss and to analyze right now, okay? We want to stop to have a break 5 minutes? Okay? Okay, let's have a break for 5 minutes, guys. Everything is okay at home?
Yes, all good, thank you.
Okay, thank you. Okay, so let's move on the excitation contraction. Let's just analyze a little bit what happened in the excitation contraction coupling from a structural point of view. We already know, we already analyzed the neuromuscular junction, but just check a little bit more deeply, okay? The neuromuscular junction is made by the motor neuron, okay? The somatic motor neuron, voluntary motor neuron that form the neuromuscular junction and allow the contraction of skeletal muscle, voluntary contraction of skeletal muscle. And as is depicted in this figure, the somatic motor neuron branches at its distal end here, forming the junction, the neuromuscular junction in the skeletal muscle fiber. This structure is also called motor end plate, okay? Neuromuscular junction or motor end plate. And here, at the end, if we analyze deeply the motor end plate in the panel B, we can see the structure of the neuron, the branches of the neuron here, that is in contact with the muscle, okay? And here we can appreciate the Schwann cell sheets all around the neuron. Why are they important? Because they allow the speed of the action potential all along the motor neuron. We can see here also we can see here the axon terminal. The red balls here is the neurotransmitter, and here the motor end plate, the mitochondrion, the vessels, the muscle fibers here with the actin and the myosin. And the important point here again is the is that the acetylcholine, in red here, is released in the synaptic cleft here, which is the process that allows the acetylcholine release in the synaptic cleft. Is the fusion, the exocytosis of the acetylcholine in the membrane. So the acetylcholine can be released in the synaptic cleft. What is important for the release of the acetylcholine? Again, that the calcium also in the motor neuron, in the branches of the motor neuron, increase. Why? Because the calcium is necessary also for the exocytosis process. Okay, we are just repeating important information that we have from the course that we already analyzed. So, from a physiological point of view, the process starts with the acetylcholine here in red that is released from the somatic motor neuron, as we shown before. The acetylcholine initiates an action potential where? In the muscle fiber. How? The acetylcholine binds to the receptor, to the nicotinic receptor in this case, on the sarcolemma of the postsynaptic membrane that is a muscle now, okay, and generate an action potential. Why? Because the acetylcholine receptor, the nicotinic acetylcholine receptor, create an open channel as soon as the acetylcholine binds to the receptor, ionotropic receptor, and the ion can flow inside. Specifically, we already talked about it. This ion is not so selective, so even sodium or potassium can go through the channel. But when the acetylcholine binds to the channel, only the sodium ion flows inside of the cell. When the acetylcholine binds to the alpha subunit of the nicotinic receptor, the sodium ions flow inside of the cell, depolarizing the muscle fiber. You can appreciate here plus, minus, and now minus, plus. So the membrane, the sarcolemma, now is depolarized because the acetylcholine binds to the ionotropic receptor and the sodium ions flows inside of a sarcoplasmatic membrane. The sarcolemma, not sarcoplasmatic, sarcolemma membrane. And the panel D just showed you the entire process. An action potential arrives to the terminal of the axon, to the branch of the axon, causing a depolarization of this presynaptic membrane. You remember that we already analyzed it, okay? Depolarization, and since a lot of voltage-gated calcium channels are present here, they sense the depolarization and they open. And calcium entry causes the synaptic vesicle to fuse with the presynaptic membrane, and the acetylcholine is released in the synaptic cleft. Every time that the acetylcholine is released in the synaptic cleft, it binds to the nicotinic receptors and sodium ions flows inside of the sarcolemma, depolarizing the postsynaptic cell, the muscle fiber. The mechanism of depolarization, this depolarization is called end plate potential, EPP, end plate potential, is always depolarizing. The muscle action potential, the end plate potential, always reach the threshold and initiate a muscle action potential. The release of acetylcholine from the presynaptic structure, from the motor neuron, always create an end plate potential and a muscle action potential. The EPP always reach the threshold and initiate a muscle action potential. What is important here? What is the important step that acetylcholine, an important role of acetylcholine here? That it creates an action potential, but in some way we need to, at the end, we need to increase the calcium also inside of the cell in order to allow the contraction. This muscle action potential, at the end, we will see how, at the end, triggers the calcium release from the sarcoplasmatic reticulum. And at the end, calcium triggers the contraction, since calcium ions combines with the troponin, etc., etc., etc. Okay, so the link between the action potential here and the contraction is the release of calcium. And now we have to see where the calcium is released and from which part of the cell is released. I would like also to remind you and to highlight that the action potential from here, from the neuromuscular junction, from the EPP, okay, moves down to the T-tubules. If you remember the T-tubules, the role of the T-tubules is to allow the action potential to move from the extracellular part of the muscle to the internal deep part of the muscle. So the action potential moves down to the T-tubules, causes calcium release from the sarcoplasmatic reticulum. Here, what is the information that we have here right now? But we just know that there is also here an enzyme. We already described this enzyme, the acetylcholinesterase. The acetylcholinesterase that metabolize the acetylcholine. And the acetyl group and the choline group can be reused okay? But the important point is that the enzyme break down the neurotransmitter, so the process stops. But at the molecular level, the transduction of this chemical signal, acetylcholine, that is converted in an electrical signal, and then this electrical signal needs to produce an increase of calcium ions. And you can appreciate in this figure that the signal is electrical here in the motor neuron, electrical here in the motor neuron, chemical here with the acetylcholine in the synaptic cleft, and then inside of the neuromuscle, inside of the skeletal muscle fiber is again electrical, because the muscle needs to be contracted. Here it's again electrical. So the point is, if the signal here, the EPP, the end plate potential here, how at a molecular level the transduction of the electrical signal is able to produce an increase of calcium signal. So at the molecular level, the transduction of the electrical signal into a calcium signal requires 2 K-membrane protein, both sensible to the depolarization. Both need to be protein that are able to understand, to feel that an end plate potential happen. And this protein names L-type calcium channels, dehydropyridine, they are called DHP, that they are mechanically gated and linked to the ryanodine receptors on the T-tubules, we will see, that are linked to ryanodine receptor on the sarcoplasmatic reticulum. I will explain you better this point, but in any case, you have to keep in mind the EPP here, the end plate potential here, that is an electrical stimuli, needs to move from the top of the muscle to the internal part of the muscle in order to contract all the muscle fiber. And at the end, to release calcium that combines with troponin to initiate the contraction. And the next slide is the entire production of a contraction of a skeletal muscle. Again, the somatic motor neuron releases action potential at the neuromuscular junction. Acetylcholine is released. The acetylcholine binds to the acetylcholine nicotinic receptor. And what happened? An end plate potential starts. A depolarization starts. You see here, a depolarization starts. So the action potential moves from the outside part of the muscle fiber till the deepest part of the muscle through the T-tubules. So, which type of ion channels should be present here in the red part, all along the T tubules. Which part of ion channel should be present here? What do you think? Something mechanically gated here or chemically gated? Sodium, but which kind of channels for sodium?
I'd say ion channel.
Which kind? Active.
Voltage gated channel.
Exactly. Voltage gated channels. Always voltage gated channels. Because the chemical signal from acetylcholine is transduced in an electrical signal. And these channels that are present here sense the depolarization occur at the end plate potential. So they start to open, like in the action potential, and allows the ions, the sodium ions, to go inside and to depolarize all the T tubules. And then potassium channel repolarize the cell, etc., etc., etc. Okay. At the end, this region, but also this region here, down, 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 are depolarized because this is the sarcolemma. And in the T tubules, there are a lot of sodium voltage gated and potassium voltage gated ion channel. So they originate the action potential here. But again, the action potential moves down the T tubules and at the end causes a calcium reticulum. Again, at a molecular level, these two proteins, the DHP and the rear channels, are voltage dependent because both of the L-type calcium channel dihydropyridine and the ryanodine receptors feel the action potential that is moving inside of the cell. The dihydropyridine sense the depolarization and move this channel, this gate, and open the gate. So the calcium here can move from the sarcoplasmatic reticulum to the cytoplasm. Let's see the next figure. The action potential arrives to the end of the T tubule. In the T tubules, other than the voltage gated sodium and potassium channel, it contains the DHP receptor, dihydropyridine receptor. And since they are voltage gated, they sense the action potential here in red. They just move a gate that is linked to the DHP receptor and open the ryanodine receptor on the sarcoplasmatic reticulum. So if I am able to open a gate in the sarcoplasmatic reticulum, obviously the calcium ions enter into the cytoplasm because they move following the gradient. So the calcium at the end binds to the troponin, moves the tropomyosin, and the power stroke can take place. So the muscle can contract. So which is the process that allows the calcium ion to increase its concentration inside of the cell? Is because the acetylcholine bind to the receptor, generate an end plate action potential, and then an action potential that move to the sarcolemma all along the T-tubules in the deepest part of the muscle, and since it is a action potential, is a depolarization, every type of channel that is voltage sensitive, every kind of protein, let's say every kind of protein that is voltage dependent, just sends the depolarization, such as the dihydropyridine, the DHP receptor, that is linked linked to the ryanodine receptor, and every time a depolarization reaches the DHP, the ryanodine receptor open the channel, and the calcium can move. So the calcium moves from the inside of the sarcoplasmatic reticulum to the cytoplasm, and then what happens? Binds to the troponin, move the tropomyosin, and the power stroke can occur. So it's okay? Okay. But again, in the relaxation phase, this is the excitation, contraction. And the relaxation, it's quite obvious. The calcium needs to decrease. And first of all, when the acetylcholinesterase deactivated the neurotransmitter, the action potential in the end plate potential does not occur because acetylcholine is not there. The action potential here is not present, so these DHP and ryanodine receptor are closing the gate in the sarcoplasmatic reticulum, and the calcium is released from the troponin and pumped inside of the sarcoplasmatic reticulum. The sarcoplasmatic calcium ATPase, the SERCA pump, the PMCA pump, the sodium calcium cotransporter, they all together decrease the cytosolic calcium and causing calcium to unbind from troponin, and muscle is released. Why? Because without calcium, the tropomyosin again cover the actin myosin binding site. So the muscle is relaxed. It's okay? Okay. So again, this is the process, okay, from the acetylcholine release in the somatic motor neuron and the calcium that combines with troponin to initiate the contraction. Let's have a question, important, okay? Sometimes I ask this question. What do you think the action potential from a motor neuron can be inhibitory? Can I inhibit a muscle contraction? What do you think? Can I have an inhibition of the muscle contraction here in the end plate potential? What do you think? You can see the answer is here, but it's also here.
Prof, is it maybe when we close the channels?
We close the channel.
Yeah, like when we are no longer having voltage, like we don't have the depolarization high enough, the channels closes.
Okay.
They're voltage gated.
Okay. Yes, but you have to block these channels. It's quite not physiological, okay? But if you block these channels here, for example, you are not going to have a muscle contraction, that's for sure. But physiologically, I mean.
Okay.
When your point is correct, but physiologically, when action potential reach the axon terminal of a somatic motor neuron, can you inhibit the muscle contraction, or it's always excitatory?
I don't know. I wouldn't know how it could be inhibited once it's started, actually.
That's a good point. You're right. But sometimes, okay, this type of synapses is always excitatory, always, because the acetylcholine in the neuromuscular junction always binds to nicotinic receptor, and they are always excitatory. Sometimes some of your colleagues, okay, says that maybe if we have GABAergic receptors here, maybe I can produce an inhibition of the muscle. But this is a wrong answer. This cannot be a excitatory, cannot be an inhibitory synapses. This is always an excitatory synapses. Okay? So, because here there is only the acetylcholine receptor. And when the acetylcholine receptor is released here, another point important is when the acetylcholine is released here, a lot of acetylcholine is released here. And a lot of nicotinic receptors are present here. Why here there are a lot of nicotinic receptors? Can you tell me why there are a lot of nicotinic receptors here? Because here, the membrane, there are a lot of introflexion of the membrane, you can see here, and this is a process that physiologically occurs when you want to have a lot of receptors. If the membrane here will be like here, a line, okay, the receptor could be 4, okay, but since the neuromuscular junction contains a lot of these structures, a lot of nicotinic receptors are present here, and even if you release a lower amount of acetylcholine, here always you are able to produce an action potential. So two reasons. Here there are in the skeletal muscle only acetylcholine receptor nicotinic is present. And second one, the motor end plate contains a high number of acetylcholine receptor, nicotinic acetylcholine receptor. So even if here is released a lower amount of neurotransmitter, always you have an action potential here, because there are a lot of receptors, and all of them can be occupied by the neurotransmitter and originate an action potential. It's okay? You understand this point? Okay. Everything is okay at home? Do you understand?
Yes, thank you.
Okay, you are very welcome. And this is the resume of the picture. So let's see this video with all the process that we analyze now.
Let's look at the structure of the neuromuscular junction before we examine how it works. The neuromuscular junction includes the enlarged distal ends of the axon, called axon terminals, and the junctional folds of the muscle fiber. The junctional folds are the region of the muscle fiber membrane, or lemma, with specialized structures for receiving signals from a motor neuron. Let's look more closely at a single axon terminal and the adjacent junctional folds. Notice that the axon terminal and the junctional folds do not touch. They are separated by a gap called the synaptic cleft. The membrane of the axon terminal contains voltage-gated calcium channels. These channels will open in response to an action potential, allowing calcium ions to diffuse into the cell. Within the axon terminal are membrane-bound synaptic vesicles that contain the neurotransmitter acetylcholine. Calcium ions will cause these vesicles to release acetylcholine into the synaptic cleft through exocytosis. On the other side of the synaptic cleft, the junctional folds contain acetylcholine receptors. The acetylcholine receptors are chemically gated ion channels that open when they bind to their ligand, acetylcholine. Outside the junctional folds, the sarcolemma contains voltage-gated sodium channels. Once the muscle fiber is stimulated, these channels open, initiating an action potential on the sarcolemma. This action potential will trigger muscle contraction.
You see, the membrane, the junctional fold contain a lot of nicotinic receptors. So the release of acetylcholine generate the end potential, okay, end plate potential. The role of the neurotransmitter. Let's see here.
Now that you know the components of the neuromuscular junction, let's examine how a nerve impulse can stimulate an end plate potential on the muscle fiber to trigger contraction. This process takes place in 6 sequential steps. Right now, our focus is on the first 3 steps leading to neurotransmitter release. In the first step, the action potential arrives at the axon terminal. To understand this step, let us look briefly at how motor neurons work. When the neuron is at rest, the inner surface of...
I stop 1 second. During the exam, if I ask you the neuromuscular junction, you have plenty of things to tell me, from the release of neurotransmitter till the muscle contraction, okay? So, keep me in mind that we have always, the question during the exam is very big question. I mean, you can talk about a lot of things. This is just one example, okay? Here you can talk about the neuronal part and the muscular part of the neuromuscular junction.
The membrane is negatively charged, while the outer surface is positively charged. This unequal distribution of charge creates a voltage across the plasma membrane. We call this voltage the resting membrane potential. The action potential that travels along the axon causes positive sodium ions to enter the cell and briefly make the membrane potential more positive. This change in the membrane potential to a more positive value is called depolarization. In the case of an action potential, the membrane charges actually reverse, so that the outside of the axon is briefly negative and the inside is briefly positive. In step 2, the action potential causes the calcium channels to open, allowing calcium ions to enter the cell. Specifically, when the action potential arrives at the axon terminal, the change in the membrane potential opens the voltage-gated calcium channels. Calcium ions diffuse into the axon terminal through the open channels because there is a higher concentration of calcium ions outside than inside the membrane. In step 3, synaptic vesicles release acetylcholine into the synaptic cleft. The influx of calcium ions during step 2 causes synaptic vesicles adjacent to the membrane to fuse with the plasma membrane, releasing acetylcholine into the synaptic cleft by exocytosis.
Let's see the next.
To understand how acetylcholine activates the muscle fiber, we need to look at step 4, in which acetylcholine binds to acetylcholine receptors. Acetylcholine molecules released from the axon terminal diffuse across the synaptic cleft and bind to acetylcholine receptors on the junctional folds. These receptors are chemically gated ion channels that open when acetylcholine binds to them, allowing sodium and potassium ions to diffuse across the membrane. In step 5, the diffusion of sodium ions into the muscle fiber depolarizes the junctional folds. This depolarization is called an end plate potential and represents a small voltage change that only occurs at the junctional folds. Let's look at how this works. Sodium ions are present in a much higher concentration outside the muscle fiber than inside. Potassium ions have a higher concentration inside than outside. In fact, the distribution of these and other ions creates a resting membrane potential, or voltage, across the sarcolemma similar to that of the axon, with the inner surface of the membrane negatively charged. When acetylcholine receptors open, large numbers of sodium ions rapidly diffuse into the muscle fiber, while a smaller number of potassium ions diffuse out. The greater diffusion of sodium ions into the muscle fiber than potassium ions out makes the inner surface of the junctional folds less negative, meaning that the junctional folds are partially depolarized. We call this depolarization an end plate potential. Finally, in step 6, the depolarization of the junctional folds triggers an action potential on the adjacent sarcolemma that spreads outwards from the neuromuscular junction in all directions. Remember that the sarcolemma outside of the neuromuscular junction contains voltage-gated sodium channels. When the depolarization of the junctional folds reaches the threshold voltage for the sodium channels on the sarcolemma adjacent to the neuromuscular junction, those channels open, allowing sodium ions to enter the muscle fiber. The diffusion of sodium ions initiates an action potential, which spreads outwards from the neuromuscular junction in all directions along the sarcolemma.
Along the sarcolemma and down into the T-tubules and in the muscle, okay? So, in the muscle, the process is this one, as we have seen before, okay? The action potential reach the deepest part of the T-tubules, open the DHP and the ryanodine receptor because they sense the depolarization. They open a gate, ryanodine receptor, a gate in the sarcoplasmatic reticulum, and the calcium moves out of the reticulum. binds to the troponin, moves the tropomyosin, and allows the power stroke of the muscle. Okay? Here is an important graph that's showing you the timing of the electrical and mechanical events during the EC coupling. Again, if I put an electrode here in the motor neuron or here in the muscle, I can record here in the motor neuron and in the motor end plate, I'm able to measure the typical motor neuron action potential with all the parts that we have analyzed, we have already analyzed, right? This is a typical action potential in a neuronal cell from -70 to +30, a little bit of hyperpolarization, and then the membrane potential return to the resting values. In the muscle fiber, I'm able to record an action potential, but the action potential is slightly different. Why? First of all, is because the membrane potential is less negative, is -90. Okay, this is an error, -90, not -80. -90. And the action potential in a muscle fiber depolarized. Why? Because the sodium channels are open. Repolarized. Why? Because the potassium channels are open later, a little bit later. And the membrane return immediately to the resting values. Here, there is not the presence of the hyperpolarization. You can see here. Okay, so it's a bit different. The somatic action potential in the upper panel here is followed by the skeletal muscle action potential measured here. But the action potential in the skeletal muscle in turn is followed by the skeletal muscle contraction. And the skeletal muscle contraction is not measured by a depolarization, but is measured by a tension, okay, a force. Okay, this is a graph where you can appreciate tension and the time, skeletal contraction and time. A single contraction relaxation cycle in a skeletal muscle is known as a twitch. Twitch is a contraction phase followed by a relaxation phase. And this period, 10 to 100 milliseconds, depends from the type of the muscle that we are studying. Okay, but a single contraction phase followed by a relaxation phase is called twitch. The last thing for today I want you to see is there is a period here in which the action potential that is followed by the action potential in the muscle fiber, but is not followed by any type of reaction in the muscle. The period called latent period here occurred at the same time at the same time that the action potential, the muscle fiber contraction, but there is a latent period in which we cannot see an electrical or a mechanical event in the muscle. And what is this latent period? It represents the time required for the electrical coupling to take place. Here, in this period, the membrane potential move along the T-tubules, open the sarcoplasmatic ion channels, release the calcium is bind to the troponin, and the troponin move the tropomyosin. After this latent period, we can start the contraction phase. But during the latent period, a lot of biochemical process occurs in the muscle, okay? So at the end, the calcium is released and the contraction phase can start. Okay? Everything is okay at home? We can stop here. Do you have biochemistry now?
No.
No?
No, we don't have biochemistry.
Ah. So you just have the lab this afternoon. Okay. Okay, guys. See you next time. See you on Monday. Have a nice weekend. Bye bye.